Good afternoon, everyone. Marcel, thank you for that very kind introduction. And thank you to the organizers and the BRCCH for the invitation to join you here today. It's a real pleasure, and I'm really looking forward to this symposium. So the organizers asked me today to speak on malaria vaccines. What I'm going to try and do in the next 20 minutes is convey to you some of the excitement, I hope, around recent advances in the field. And then the second part of the talk, try and convey to you at least my thoughts on what are the future challenges for this field. To begin with, though, just to orientate us on malaria, I'm sure most of you know malaria remains a huge burden on global public health, with around 200 million clinical cases still every year. And despite fantastic progress with malaria control measures, we're still looking at around 500, 600,000 deaths every year, primarily in young children in sub-Saharan Africa. And that, for me, is one of the biggest arguments for why we still need to strive to develop a vaccine for malaria. Most of you may know malaria is caused by Plasmodium parasites. This picture here shows you their complex life cycle as the parasite shuttles between the human host and the mosquito vector. Complicated process initiated by the bite of an infected Anopheles mosquito. This releases a form of the parasite into the skin called a sporozoite. These migrate rapidly from the dermis of the skin into the liver, where they infect hepatocytes. And over a period of about a week, they then develop into a new form called a merozoite. These rupture out into the blood and begin to undergo exponential growth in the blood. And it's that blood stage that leads to the clinical symptoms and the associated morbidity and mortality. During that time, some of those cells form sexual gametocytes. These go back up into a mosquito in its blood meal and develop over a couple of weeks back into sporozoites. So the point of going through that is to orientate you around the life cycle, but just to convey the sheer complexity as this single-celled, shape-shifting animal moves through its life cycle. It's important to note there are in fact 6 species of Plasmodium that infect humans. So when we talk about elimination and eradication, we need to think more widely. But in the context of this talk today, I'm going to focus on Plasmodium falciparum, which causes the vast majority of those deaths that I referred to. Now, vaccines mediate protection via the immune system. So before we get to vaccines, I'm just going to touch briefly on naturally occurring immunity to malaria. This is what happens when you're naturally infected in the field. And unlike a number of pathogens where we're accustomed to catching it once and then developing an immune response that protects us for a long period of time or perhaps even life, malaria is very different. Some of you may know that immunity acquired to malaria is non-sterilizing, as in you can catch the infection over and over and over again. It's slow to acquire. It requires repeated infection. And in short, without delving into immunology, it's complicated. It looks like the immune system, the antibody response, and the T-cell response both play a part. And this leads to what we call anti-disease immunity, whereby you stop getting sick and inflamed, but also antibodies that control the level of parasites in the blood. And this graph here on the bottom left shows you what really happens when you catch malaria for the first time. You're at very high risk of severe disease, shown there in the green bar. But the more you catch it, you transition to mild disease in the blue. And by the time you become an adult, you're an asymptomatic carrier. And we believe the majority of that immunity is mediated against the blood stage parasites, shown there in the green box. So what about vaccines? Well, as many of you know, malaria has been particularly challenging to develop a vaccine for, and I reflected on what the challenges the field has faced in recent years, and I listed them, or what I thought were the main challenges here. People have struggled to identify antigens or targets for the vaccine. The parasite has over 5,000 proteins we have to work through in order to identify those. Very different to, say, SARS-CoV-2, where you just have one spike protein on the surface of the virus. And in hunting for those antigens, you ideally need to find ones that are conserved. That means you get broad antibody responses not ones that are strain specific. You've got to cover genetic diversity. You ideally need the antigen to be essential. That means it's so important to the parasite, it hasn't got a backup system if you target it. And even if you can tick those two boxes, you then need it to be immunologically susceptible. If you target it, the immune system's got to be able to stop the parasite. Alongside that, the immunology is important, what we call immunological correlates or immune responses that we think associate or predict protection. And we've had to sort out that immunology, otherwise as a vaccine developer, you're flying blind in terms of what you're trying to achieve. And it's also become apparent that for malaria at least, you need really strong immune responses. Now, of course, this is all relative, but they're at the real high end of the spectrum if you're going to stop this parasite as it shuttles around that complex life cycle. So we've needed technologies in humans that are safe and deliverable and deployable and scalable, but also ones that give us these very strong responses. So what about the advances in terms of vaccines? Well, there's a number of strategies now that are showing promise around the life cycle. They're shown here. At least 3 of them involve antibodies that stop infection with the parasite, either the sporozoite getting into the liver, the merozoite into the red blood cell there at the bottom, or antibodies that interfere with sexual development inside the mosquito. And it's also possible to induce T cells that act in the liver and kill the infected cells. What's interesting is that none of these strategies, in fact, mimic the natural immunity that I was talking to you about just a few moments ago. All of these strategies at the moment are seeking to block infection and are not responses that you naturally acquire when you're repeatedly infected. And for the purposes of today's short talk in the next 15 minutes or so, I'm really going to focus in on the 3 opportunities there that involve antibodies at the sporozoite stage, in the blood, and at transmission. And in recent years, it's important because we've identified antigenic targets that I was just talking about, and importantly, they've now been validated, including with monoclonal antibody studies. So the immunology is getting a lot stronger in terms of understanding what we need to achieve. So we've got targets, the immunology is greatly improved. What about the vaccine technologies and getting these very strong responses? Well, I've listed here on the slide what I would say is the hierarchy with which different technologies can induce antibodies. So we've been working on this for a number of years, and at least in our hands, when we use gene-based delivery platforms, such as DNA or viral vectored vaccines, which you may be familiar with from the COVID field, we do get antibodies, but they're relatively at the weaker end of the spectrum. But with recombinant proteins or virus-like particles, so-called VLPs, you can get much stronger antibody responses. And so we've been focusing on those. In the case of the VLP, you array the protein on the surface of a virus that clusters the B cell receptors, and you can activate the B cells that make the antibodies more easily. There's, of course, a lot of interest with mRNA, given progress in the last 2 years with COVID. We've not tested them yet for malaria, but based on data from COVID, I suspect they would fall at the higher end of the spectrum. It would be exciting to see what happens with those in the future. Of course, with recombinant vaccines, you also need an adjuvant to activate the immune system. We're lucky in malaria that we've had access to some of the strongest adjuvants to give us these very high responses. Those include AS01 from GSK and Matrix-M from Novavax, both saponic-based adjuvants. And another, I think, positive advance for the field is now the engagement of high-volume, low-cost manufacturers, particularly those in India. They're engaging, taking on these new technologies, and I think that offers hope for the production of these new vaccines to scale in the future. So what about specific examples? Well, starting with the sporozoite, this protein called CSP covers the surface of the sporozoite. It has these 4 amino acid repeats in the middle of the antigen. And the vaccine you may have heard the most about, RTSS, targets this protein. It's been developed by GSK over the last 30 years, given in their strong adjuvant AS01. So this is a hepatitis B vaccine, the Hep B particle with a bit of the CSP all over its surface. And it induces antibodies that block infection of the liver. So this vaccine has completed phase 3. It's been through pilot implementation studies and received a positive approval from the WHO this time last year for use in areas of moderate to high transmission. The headline efficacy result was 36% against clinical malaria over 4 years. More recently, a vaccine based on the same concept has been developed by Oxford, Serum Institute of India, and Novavax called R21, delivered in the matrix adjuvant. It's anticipated in the future this vaccine could be more scalable than RTSS, at least in the short term. It's currently in a phase 3 trial, and in recent data has shown high-level efficacy, but it's important to note that this is over a malaria season, and it's about 77% efficacy in the first year. They then boosted again and got the same result in the second year. So these new data with R21 at least showing how with these types of vaccine we can get relatively high-level efficacy over the short term in a malaria season, but of course that is requiring annual boosting. Coming up behind these, exciting developments with monoclonal antibodies, these developed by Bob Cedar at the NIH. He's recently published 2 studies where they got very high level of efficacy in a human challenge study. These antibodies targeting the CS repeats at the bottom, and they're now in field trials. And I think one to watch here, because potentially offering a single shot intervention that could perhaps provide protection, again, over a malaria season or up to a year. On the blood stage front, so moving around the life cycle now, there's been a lot of excitement in the field with the identification just over 10 years ago with a protein called RH5. This is a conserved antigen that forms an essential interaction with a protein called pathogen on the red blood cell. This is what it looks like here in the middle. It has a diamond shape binding to its receptor, and it's also released in a conserved complex. Here we've been able to assess antibodies against RH5 that we hope block that interaction and block infection using what we call a growth inhibition assay, or GIA. It's very similar to a virus neutralization assay. We culture parasites in blood, add in purified antibody, and look at inhibition of growth. An exciting result for us was that we were able to show here on this graph with the assay on the x-axis and protection on the y-axis that it's a very good predictor of vaccine protection in a non-human primate model. And we were able to validate that again with monoclonal antibodies. And from these data, we were able to identify a level of antibody that at least correlates with protection in monkeys. So we've spent a number of years now undertaking a series of clinical trials trying to reach that level of antibody in humans. These have been done in Oxford, led by Angela Manassian, and at the Ifakara Health Institute in Tanzania, led by Ali Alotu. And these data just show you a spectrum of the antibody responses that we've achieved. Just to highlight, this is where we began about 6 years ago with our first trial in the UK. We've been very excited by our most recent data, where we're now able to get antibody levels 70 times higher. And for the first time ever, we've crossed that threshold we wanted to achieve in humans. So this vaccine at the blood stage will now go forward to field efficacy trials. And then finally, just coming to the transmission stage, again, this part of the life cycle has a very good assay called the standard membrane feeding assay, whereby we can add in antibodies and look if they inhibit development of the parasite inside the mosquito. And there's a number of antigens which have got good proof of concept in the lab. So here you can see in this experiment in the controls, you're getting the parasites developing in the mosquito. But if you add in antibodies against these targets, you can completely block development of the parasite inside the mosquito. So I think there's some exciting data coming from the NIH group. We've got a vaccine against this PFS 230. More recently, the group in the Netherlands have validated again with a monoclonal antibody against one of these antigens, showing that you can induce this effect by passive transfer in humans. So a lot of advances, I think, in the last few years. It's been an exciting space. Important to note these different vaccines, subunit vaccines are at different stages of development. The RTSS and R21, the most advanced, but coming up quickly behind them are these new vaccines around the life cycle at the blood stage and the transmission stage, and also at the monoclonal antibodies. So I began the talk reflecting on the challenges, perhaps now of the past. This was the slide that I used. And I think there's been a lot of exciting progress. So just to finish, what about the future challenges that are coming? Well, I reflected on this before making the slide. I think there's a few that are still ahead of us in terms of this space. Looking ahead, it's going to be important to define now how we deploy these first generation antisporozoite interventions. There's interest in how we use them with either seasonal immunizations, so timed with the malaria season, versus vaccinating all year round. We have monoclonal antibodies coming onto the scene, that may be single shot annual interventions. And I think it's also important for us to reflect on the fact that these interventions will block infection. So what's going to happen 5 years down the line? We may end up with slightly older children who've had less exposure to malaria and have acquired less natural immunity. The risk is you may see rebound of severe disease in that population in the future. We hope we don't, but I think it's important to start reflecting now on what we may anticipate in the future. I think another exciting challenge is that we now have the opportunity to assess and combine and utilize multi-stage interventions. I think we now need to put our mind to how we will bring in these other components around the life cycle. And the other big challenge for us is whether we can in fact develop longer lasting immunity by vaccination. We seem to be reaching there at the peak, but the responses are waning over time. So I'm just going to finish by reflecting on that last point. And this is really relevant to all antibody-inducing vaccines for malaria. We can't rely on natural boosting because, as I said to you, these targets are not dominant in the natural response. So this is a normal antibody curve. They go up and then they come down over time. They decline quite rapidly, and then you go into the memory phase. And you need to imagine at a certain level, this is totally arbitrary, above which you are likely to be protected, but as the antibodies decline, the vaccine efficacy will wane. And of course, with annual boosting here, the attempt is to get you back up over that line every year by re-administering the vaccine. Now, I don't want to pick on RTSS. This is common to all of the types of vaccine, R21, our RH5 vaccine in Oxford. The responses go up and come back down. The question is, can you shift that curve? How do you overcome the apparent normal or natural antibody response curve? Well, there's a few ideas. You could keep shifting the curve up, getting responses higher and higher and higher. I wouldn't argue with that, but we're already using state-of-the-art technologies, and you should never say never. But at the moment, we're already really high, so I think it's going to be a massive challenge to get the responses even higher. What about changing the shape of that curve? Well, we got some interesting data from a recent trial where instead of giving the vaccines at monthly intervals, 0, 1, and 2 months, we delayed the last shot and lowered the dose. And these were our data in UK adults. You can see in purple, the responses came up, but then declined more slowly over time. And 2 years later, they were 10 times higher than the other regimens. So this is modulating regimen and dose, and I think it's really been underappreciated in the past. And I think going forward in the future, we need to be a lot more adventurous and ambitious in our phase 1s because there's potential for big gains in terms of longevity. And then finally, the other possibility is that we lower the bar, achieve protection with a lower level of antibody, which means it will last for longer. And here people are really focused on improving the quality of the antibodies, so they're more functional, you can kill the parasite more efficiently. And the hypothesis really is that when you make a polyclonal antibody response by vaccination, you get lots of different clones, that you can enrich the response with the best antibodies. Now, lots of us are trying to do this. It involves a lot of structural biology studies, aligning structure with function. But just to show you some proof of concept, at least in animals, these are 2 types of vaccine we've made for the blood stage in Oxford. The one in black is our current one in the clinic. The one in blue is a new one we've just produced and will enter the clinic next year. But here we've plotted parasite killing against antibody. And you can see with the new vaccine in the blue, we've shifted the curve to the left. We're killing the parasite with 10 times less antibody. So we're crossing fingers we get the same result in humans, but just to give you a flavor of the things people are trying. And then finally, The other possibility is to look at these multi-stage approaches, combining vaccines around the life cycle, or potentially even vaccines and monoclonals. The hope is they might synergize, and by that I mean each vaccine makes the other work better, and we end up with a better efficacy result. The fields talk about that a lot. The data are relatively weak on the concepts at the moment, but it really relates to what I call force of infection around the life cycle. So if you have an anti-sporozoite vaccine, the hope is less sporozoites go into the liver. I mean, you're aiming for none, but the aim is to reduce the number going in. The question is then, would a vaccine that works in the liver be more effective because it has fewer infected cells to clear? If less parasites come out of the liver into the blood, is it easier for a blood stage vaccine to protect? If a blood vaccine reduces the number of sexual gametocytes, is it easier to block transmission? And if less gametocytes go into the mosquito, you get less sporozoites coming out, and so on. And I think this is a really exciting space because these questions haven't been addressed. I think they're a challenge for the future, and should offer some really exciting angles for us to explore in terms of improving vaccine efficacy. So I will leave it there. I will just acknowledge the team in Oxford. I showed you some of their data during the talk, as well as the wider community whose work I've been referring to. And thank you to the funders, and finally you for your attention. Thanks very much.